It's a true pleasure to introduce Robert Weinberg, much better known as Bob. Bob has made many seminal contributions to cancer research, as evidenced by the fact that people don't have a place to sit. Among his many firsts, he was the first to show that human cancer cells contain activated oncogenes as exemplified by mutant RAS that, when transferred into normal cells, can endow upon them cancerous properties. He was also the first to clone and functionally validate the tumor suppressor, in this case B, carcinoma gene. But perhaps, in addition to his many experimental breakthroughs, and perhaps not less important, is his role as a thought leader in cancer research. And his many reviews and lectures have greatly shaped the way we look at cancer now. We, the whole cancer community, cancer research community, look at cancer and understand cancer. We ask questions about cancer. Perhaps less be known to this audience, but maybe of, again, equal importance, Bob did a postdoc at the Weizmann Institute. And for all the postdocs in this audience, this is cheer up, because one day maybe you can be the next Bob Weinberg. So without any further ado, Bob is going to tell us about epigenetics and cancer biology. Please, Bob.
Thank you. Ani Motsameach Liot Kan. I worked here as a postdoc in 1969-70. Weizmann. And every time I come here, I'm reminded how unfair life is. Because when I came here, I was postdocing with Ernst Winokur. And I've changed a lot in the last 49 years. Ein Hevdel, he looks exactly the same as when I started working with him. So, as JFK said, life is unfair. We don't, and Kavod, Lihot, Al-Sela, nice to see you. They're both Kimat Ben Tishim. I'm not going to reach that age, but every day is a gift. B'chol ofen, thank you for coming. Are you the technician here? It's not working. Okay, well, okay. I think we're okay, yes? No?
You'll be okay now. Now you're okay.
Okay. Well, I'm always reminded of the phrase, Yiyeh Tov, when things are worse and everybody knows things are going to get worse even. Yiyeh Tov. So I'm very pleased to be here. I'm flattered at this large audience. And I'm coming here to talk today about the role of epigenetic programs in tumor progression. Our thinking is greatly influenced by the work of Kinsler and Fogelstein in 1989, who first demonstrated. Can you hear me when I'm aiming like this? Can you hear me? You're the most important people. Good. I'm always reminded of the work of Kinsler and Fogelstein, who for the first time enunciated the idea that multi-step tumor progression depends on the acquisition of a series of somatic mutations, here, loss of tumor suppressor genes, and the acquisition of an oncogene that empowers cancer cells to grow as primary tumors. The focus on these mutations, these genetic changes, has distracted attention away from an equally important and potentially even more important source of cancer cell phenotypes. And these are non-genetic changes in cancer cells. Here's one example of one of these epigenetic programs. Here's a recent work that was published, which shows that in a genetically engineered model of small cell lung carcinoma, if one activates oncogenes in one cell type or another cell type, the resulting tumors have a great difference in their transcriptomes, even though they came from very similar cell types, but distinct cell types. And that impresses on us the influence of the normal cell of origin, what operates here in normal cells, and continues to operate throughout the entire process of tumor pathogenesis. It is not as if the oncogenic lesions that are acquired through somatic mutations eradicate the transcriptional program that pre-existed here in the normal epithelium. I want to focus on a later stage of tumor progression over here, because here, as I will argue, this last step of tumor progression is also fully or highly epigenetic and does not depend. Can you hear me over there in the back? Anybody out?
Yeah.
Good. Also does not depend on genetic alterations in cancer cells. Indeed, people who've tried to find mutations that enable carcinoma cells to metastasize have repeatedly failed to find such mutations, which begins to suggest that there are other mechanisms at work. So here is at least two sources of non-genetic variation which strongly influence the behavior of cancer cells. This last step is actually very important because 90% of cancer-associated mortality derives not from primary tumors, but instead from the metastases that derive from these primary tumors. And this step from here to here has been called the invasion metastasis cascade by Josh Yishiabu-Fidler, who indicated that there are actually 5 or 6 distinct steps that operate during the dissemination of primary cancer cells. First, local invasion, invasion of the blood vessels, translocation, extravasation out of blood vessels, the formation of micrometastases, and the final step of colonization, where micrometastatic cells succeed in growing into macroscopic metastases. This process is extraordinarily inefficient, and the greatest inefficiency happens here when cancer cells that have landed in a foreign tissue have to figure out how to make a living in a foreign and potentially inhospitable tissue. And the question is, how do cancer cells acquire all of these distinct phenotypes, these distinct capabilities? Much of the answer to that, perhaps not all of it, derives from this biological phenomenon, which I will call the epithelial-mesenchymal transition, where we see here epithelial carcinoma cells expressing cytokeratins. Out here is the stroma, the recruited normal stroma, and these green cells derive from the epithelial carcinoma cells, but have acquired a mesenchymal marker, Weizmann, and in fact are phenotypically highly mesenchymal. And here you can begin to see that this switch from an epithelial to mesenchymal state is stimulated by signals that carcinoma cells get from nearby stromal cells. Here you see the outside stroma, here epithelial cells, and here you see a rim of carcinoma cells that has shut down cytokeratin and instead now express Weizmann. Once again, these cells are in direct contact with the stroma, and that begins to suggest that the activation of this program occurs in response to contextual signals that these cells receive from the nearby stroma. Here's some examples of this. Here's the work of Ann Lee, who demonstrated that carcinoma cells that release interleukin-1 that impinges on cells in the stroma, fibroblasts and mesenchymal stem cells, and evokes in turn a response with multiple distinct cytokines being released that ultimately activate in the carcinoma cell that initiated this bidirectional intercommunication, the activation of the previously latent EMT program. Here's another example of these heterotypic interactions between two distinct cell types. Here's a carcinoma cell that has already activated its endogenous EMT program, and here we find that through juxtacrine signals with monocytes and macrophages in the stroma, this interaction leads ultimately to the expression of an EMT-inducing transcription factor called here TWIST, which reinforces and stabilizes and maintains the already initiated EMT program. And there are clearly many other types of heterotypic interactions in which stromal cells activate previously unexpressed EMT programs in carcinoma cells. Here's a manifestation of the EMT program early in embryogenesis, where in the sea urchin embryo, we see that cells that have undergone gastrulation release these mesenchymal and endodermal cells having undergone an EMT. So this is a very ancient lineage. And indeed, this program, having been described by Elizabeth Hay already 30 years ago, is a multifaceted program, a cell biological program, which has profound changes in the phenotype of the cells, as you can see here. There are as many as 400 changes in gene expression that occur when cells activate this previously latent program. And if you look here, you see that some of these phenotypes are precisely those one associates with the carcinoma cells of high-grade malignancy. In fact, I've recently tried to help develop a consensus among EMT researchers as to what an EMT program is. And this is a most frustrating undertaking because cell biologists have one point of view, and the biochemists have another point of view, and the signal transducing people have another point of view. All the time there's a vikuhim between each point of view, and what turns out to be the case is that there is a central EMT program that is shared in common and has a relatively small number of changes, reduction in ecadherin, activation potentially of vimentin, maybe one other change, but from there on the EMT programs diverge in a whole series of different directions. So EMT program is really EMT programs, and each tumor may have its own distinct EMT program, its own distinct version thereof. The EMT program is choreographed in cells by a set of transcription factors. Here you see them, and you see how they're expressed in a whole series of metazoan embryos, snail, slug, twist, and so forth. And these images, together with the information that these transcription factors are encoded in the genomes of all metazoa, indicates clearly that the EMT program was already invented maybe in the Cambrian era 600 million years ago, and has been maintained very conservatively in the intervening time, simply because it obviously plays a critical role in normal development on the one side, and even in wound healing on the other side. Here we see one manifestation of this. This is a polyoma middle T model of adenocarcinoma of the mammary gland that Chinier undertook, and what you see here are ecadherin positive cells. They're highly epithelial, and here you see that the snail EMT transcription factor is suddenly expressed here in this tissue, and in that area the ecadherin expressing tumor cells are eliminated. They've lost ecadherin. And here you see the inception of the EMT program early in tumor pathogenesis, and one demonstration, and there are hundreds, of how the EMT program can shift a critical cell biological molecule, ecadherin, which is responsible for the lateral interactions between adjacent epithelial cells. Another aspect of the EMT program is indicated here, starting with the work of Muhammad Al-Hajj and Michael Clark, who showed in 2004 that if one depicts the hierarchy of stem cells and non-stem cells in a tissue, then in fact within cancers there also exists such a stem cell hierarchy, with using their markers CD44 and 24 in this FACS analysis. The non-stem cells are here, and up here reside the stem cells, although we have no reason to believe that either of these populations is totally homogeneous. Nonetheless, this represents a very powerful experimental tool, and here I refer to the work of Sender Mani and Wenjun Kuo, where they asked the following question, and to this day I have no idea why they asked this question, but they did. What's this intersection between the EMP program that I discussed before and the stem cell hierarchy that I just mentioned now? Is there any connection at all? To me this seemed like a bizarre question, but they moved ahead anyhow. And of course I only heard about these experiments weeks and months after they were done. They took snail, an EMT-inducing transcription factor, EMTTF, or twist, another EMT transcription factor, and they introduced each on its own into these non-stem cells, asking whether that would have any effect on the overall behavior of these cells. And what happens is the cells, as you can see here, underwent a morphological transition from an epithelial cobblestone phenotype into a more mesenchymal phenotype. And even more importantly, the cells migrated in the presence of either snail or twist the non-stem cell into the stem cell position. Again, I mentioned that this position at the top left is hardly a homogeneous population of cells, but it began to suggest the notion, which has been borne out repeatedly in the years since. Also, this work occurred in the work of Alain Cuisseau at the same time in Lyon, that there is an intimate connection between the EMT program in certain epithelial tissues and the stem cell program, for reasons that to this day I don't understand the purpose, the teleology. Here's work that Weizmann undertook just to demonstrate that this intersection between EMT and stemness also operates in the context of normal epithelial tissues. Here, he took mammary epithelial cells and implanted them in the mammary stromal fat pad. And if one does that, and there are mammary stem cells in that population of implanted cells, one can generate an entire mammary ductal tree, which is phenotypically essentially identical to a normally developed mammary ductal tree. And so here he took 10,000 cells from the adult mammary gland, implanted them, and nothing happened. In these cells, here he expressed transiently these two EMT-inducing transcription factors that operate coordinately, and when he did so, as few as 100 of these cells that many weeks earlier had transiently experienced the actions of these EMT-inducing transcription factors, here as few as 100 of these cells actually now acquired the ability, upon implantation in a cleared mammary stromal fat pad, to produce an entire mammary gland, indicating that they indeed had acquired stemness in the context of normal epithelial biology. Here's the work of Chimier, still unpublished, in which he began to ask the question, in which tissues is the slug EMT-inducing transcription factor expressed? Indeed, here you see the emphasis on slug because she found it to be expressed strongly in the basal cell layers of the normal mammary epithelial ducts. And here she's begun to examine in which tissues, normal tissues, does one have this EMT-inducing transcription factor actually expressed. And to date, it seems it's expressed in all bilayered epithelial tissues throughout the body. But to date, she and we don't understand what happens in single-layered epithelia. So there is some generalizability of what I'm talking about today to a whole series of epithelial tissues throughout the body, but not all tissues. We don't really understand whether there is actually an association between the EMT program and stemness in these other tissues. I suspect there is, but it is still undemonstrated. Here's yet work of Chimier, in which he asked in a model of mammary adenocarcinoma pathogenesis, what about the presence of cancer stem cells? And one measures these stem cells by implanting a population of cells in a host mouse at various dilutions and calculating what's the representation of tumor-initiating cells, tumor-seeding cells in those tissues. And one can calculate that in cells that express slug in the carcinoma cells, 1 in 170,000 cells could initiate a tumor, whereas those that express its cousin, its paralog snail, 1 in 91 cells could do so. So there's about an 800-fold difference in the representation of tumor-initiating cells here, indicating that snail is important actually in adenocarcinoma pathogenesis, and its cousin, slug, is, as I showed you before, important in normal mammary stemness. So these two paralogs of one another, they've been paralogs for 600 million years. One is responsible for normal stemness, and the other is responsible, at least in this model of breast cancer pathogenesis, for the development of cancer stem cells, tumor-initiating cells. And there are many echoes of this in the literature in which other types of carcinoma cells have been found to acquire stemness as a consequence of having activated their previously silent EMT program. Again, for reasons that we don't really understand. Here's yet another demonstration of the powers of the EMT program. This is the work of Zuzana Keshkeshova and Yasmin Nikok, who took weakly metastatic human breast cancer cells, they're called MCF7 RAS, and implanted them in a host mouse and waited for 10 weeks, let's say. And these cells, unmodified, produce localized tumors with a very smooth edge, indicating that they're non-invasive. However, if they turned on this pair of EMT-inducing transcription factors, I just showed you them in the context of a normal mammary gland, and turned them on briefly and then shut them off and waited another 10 weeks, the tumors that resulted 10 weeks later remembered that they had been exposed to these powerful EMT-inducing transcription factors, continued to express highly malignant phenotypes, including, as you can see here, the invasive edge where carcinoma cells are invading into the nearby stroma. Perhaps more persuasive is this measurement of the number of micrometastases and macroscopic metastases that are generated in the lungs of such mice. Normally, one gets almost none, but cells which 10 weeks earlier had experienced slug and SOX9, these two EMT-inducing transcription factors, briefly for 2 weeks, now they can produce 100 macroscopic metastases and as many as 400 microscopic metastases. And this is the beginnings of a proof that turning on the EMT program in primary cancer cells is sufficient to enable cells to physically disseminate from the site of primary tumor formation to a distant tissue. Whether this happens in all tumors is not clear, but if this is borne out, this indicates that the process of physical dissemination is entirely an epigenetic process, where cells that are mutated to form primary carcinomas already have the genetic endowment to disseminate physically, but only require activation of their EMT program in order to complete all of these steps. That is, they don't need additional mutations to enable or to empower metastasis to occur. The still unanswered problem is right here, because there can be thousands, maybe even millions of these micrometastases, but only on rare occasion do the cancer cells figure out how to make a living in a distant, unfamiliar tissue, which doesn't offer many of the cytokines and extracellular matrix constituents that create the microenvironment in a normal tissue. And so this remains an area of active research. How this happens, we still don't understand. Still, this represents a very simple conceptual solution to the problem of the invasion metastasis cascade, because one can argue that these multiple steps all fall under the control of the EMT program, and thus don't require a whole series of distinct programs or distinct mutations in order to reach completeness. So this represents a major challenge, and by the way, the EMT can also expedite extravasation, escaping from a blood vessel, as well as proliferation, brief transient proliferation after carcinoma cells have escaped a microvessel and attempt to initiate a program of proliferation in the parenchyma of a distant tissue. Here is yet another consequence of the EMT program, and this is by now an old slide I received from John Haley, where he began to examine the effects of the EMT program on the responsiveness of carcinoma cells to chemotherapeutic. And what he found was that cells that were epithelial had a much higher rate of death than cells that were more mesenchymal. And this is only one of literally dozens of reports which indicate that activation of the EMT program confers a measure of chemoresistance on carcinoma cells, rendering them unresponsive to a variety of chemotherapeutic treatments. So one can begin to add yet other phenotypes to the list of phenotypes that are conferred by the EMT program. There are these two, which obviously are very important. Self-renewal is important because when a cancer cell disseminates to a distant tissue, its ability to serve as a tumor-initiating cell is critical to the subsequent success of informing a colony of cancer cells at a distant site, i.e. so that it can successfully be a tumor-initiating cell. Here is more recent work of Anushka Dongre, in which she asks the following question. Is there any effect of cells moving from an epithelial to a mesenchymal state in terms of the responsiveness of these carcinoma cells to elimination by the immune system? These days, you cannot give a seminar in biology unless you talk a little bit about checkpoint immunotherapy. Otherwise, people snicker and walk away in the middle of your seminar. So here's my contribution to checkpoint immunotherapy. In the old days, it was microarrays, and before that, alternative splicing. So the meal of the day is checkpoint therapy. And here is my contribution. She and Brian Beer developed cell lines from the polyoma middle T model of human adenocarcinoma of the breast pathogenesis. And they developed two pairs of cell lines, an epithelial and mesenchymal cell line prepared in one way, an epithelial and mesenchymal cell line prepared in another way. As you can see here, for example, these epithelial cells express E-cadherin, which is the hallmark of the epithelial state. The mesenchymal cells express a whole series of mesenchymal markers, and so they bred true. And I will tell you that somehow these cells maintain their residence in these two distinct alternative phenotypic states. The same is true over here. And here we can see an example of the maintenance of residence in one state or the other. Here, the epithelial carcinoma cells, when they form tumors, when they're implanted, continue to express E-cadherin, but don't express Weizmann, which is a mesenchymal marker. Conversely, the so-called mesenchymal cells don't express E-cadherin, but they do express Weizmann. So there is a retention of residence in one phenotypic state or another for reasons that we don't totally understand. That is to say, we don't understand what stabilizes and maintains the long-term residence of these cells in these alternative phenotypic states. Here's the beginnings of work of Anushka Dongre in trying to understand the interactions of these two distinct cancer types, which are, as far as we know, derived from common tumor and are genetically essentially identical. Here are their interactions with cells of the immune system. As you see here, here's one pair of cells, double-headed arrow, another pair of cells, double-headed arrow, and the tumors that they generate. And as you can see here, in terms of cytotoxic T cells, the epithelial carcinomas that form are full of cytotoxic T cells, whereas the mesenchymal counterparts have far fewer cytotoxic T cells. And this is true for both pairs of cells. In other words, the epithelial cells are invaded with highly potent cytotoxic T cells that should, in principle, be capable of killing them. If one looks at the state, the biological state of these invading CD8 cytotoxic T cells, one finds that in the mesenchymal tumors, here in blue, the cytotoxic T cells show markers of what's called exhaustion. That is to say, they've lost many of the markers that make them functionally active, and even though there may be some cytotoxic T cells in the mesenchymal tumors, those cytotoxic T cells that are present are functionally relatively inactive, if not totally inactive. On the other hand, you see here the fact that if one looks for a marker of exhaustion, which means these cells are really tired, or in the language of immunology, exhausted, then the mesenchymal tumors do harbor, as I just mentioned, cytotoxic T cells, but they are essentially inactive and unable to launch an attack on the carcinoma cells. Here one sees one other actor in this drama, because as you may know, the regulatory T cells, which are defined by this marker, these regulatory T cells operate to suppress the actions of cytotoxic T cells as well as NK, natural killer cells. And what you see is that the mesenchymal tumors in both cases have far higher numbers of these regulatory T cells whose function it is to suppress the actions of the cytotoxic T cells that otherwise would launch an attack on the tumor. Here is yet another difference in these two kinds of tumors, because here one looks for immunosuppressive macrophages. As it turns out, there are at least two major types of macrophages. There are the M1 and the M2 type. M2 are immunosuppressive. There are people in the macrophage community that argue there are really 10 or 20 different kinds of macrophages, and they argue very heatedly about this. And we always have to remember the reason why academic disputes are so nasty and bitter is that the stakes are so low. So here we see a situation where here we see immunosuppressive macrophages that are present in far higher concentrations in the mesenchymal tumors than they are in the epithelial tumors. Here's yet another demonstration of the recruitment of immunosuppressive macrophages into tumors, because the mesenchymal tumors acquire large numbers of these immunosuppressive macrophages, which they may well use to suppress attacks, to blunt attacks on the carcinoma by, let's say, the cytotoxic T cells. It's a very different immunological environment. And here, if we look at the localization of these macrophages, these immunosuppressive macrophages, they're held at the outside of these epithelial tumors. They are not allowed to come in. But in the case of the mesenchymal tumors, the macrophages, the immunosuppressive macrophages, are all over the place, suggesting, but hardly proving, that they can act here to protect the tumor from attack by cells of the immune system. And here's yet another quite stark difference, and that is the expression of MHC class I proteins on the surface of these carcinoma cells. As you can see here, for example, when carcinoma cells pass from an epithelial to mesenchymal phenotype, the levels of MHC class I proteins decrease as much as 100-fold, moving from one state to the other. And as you may know, as you surely know, these MHC class I proteins are important to display on the surface of cells oligopeptides that inform the immune system as to the state of the biology of the cells. These are externalized oligopeptide antigens which serve as attack targets for cytotoxic T cells. And they're almost absent in these more mesenchymal carcinoma cells, relatively speaking. So this suggests also an immunoevasive maneuver. Not only do the carcinoma cells actively suppress immune attack, but they're also highly evasive in that they no longer display the cell surface proteins that are critical to alert the immune system to the presence of alternative and aberrant proteins within the carcinoma cells. Here's an amusing difference, and this one gains me respectability in the community of tumor biologists. Here we do checkpoint immunotherapy, finally. Most people come to this seminar, all they care about is checkpoint immunotherapy. Perhaps, maybe in Yitzhak Hashem, you'll be interested in other aspects of this. But here we see the response of the epithelial carcinoma cells to checkpoint immunotherapy, imposed in this case by anti-CTLA-4. And here we see how these epithelial tumors, they normally grow out, but if they're given checkpoint immunotherapy, then their growth is blunted, is greatly reduced. On the contrary, the mesenchymal tumors grow out normally like this, and if they're given checkpoint immunotherapy, if their hosts are, nothing happens. So they're actually non-responsive, unresponsive to checkpoint immunotherapy. And here we see an amusing experiment in which Anushka Dongre mixed epithelial and mesenchymal cells in a proportion of 1 part mesenchymal and 9 parts epithelial. The question here was, could the minority of mesenchymal cells in a tumor protect the tumor as a whole, which was 90% epithelial, from attack by checkpoint immunotherapy? Here you see that the mesenchymal cells are resistant. I've showed you this before. The epithelial cells are sensitive to checkpoint immunotherapy, and this 9-to-1 mixture almost totally protects the tumor as a whole from attack by checkpoint immunotherapy. And then it can be added to the growing literature. There are only hundreds of new papers each week on checkpoint immunotherapy, because although one has described its successes, and they are clear and dramatic in certain kinds of tumors, such as melanomas, the fact is that the majority of carcinomas don't really respond to checkpoint immunotherapy, at least they haven't to date. And that begins to suggest that one has to consider aspects of the biology of the targeted tumors, such as, in this case, the activation of EMT programs, which occurs frequently in clinical carcinomas. So this is not irrelevant, because here one sees a 10% minority of more mesenchymal carcinoma cells within a tumor, cells that have undergone an EMT, already protects quite well the tumor as a whole from attack by the CD8 T cells, the cytotoxic T cells of the immune system. Here is yet another aspect of the EMT program. This is the work of Brian Beery, who demonstrated that within a given tumor, one sees cells of all phenotypes, from epithelial to mesenchymal, coexisting within a tumor. This may be more heterogeneous than most, but it begins to indicate that there's a whole variety of intermediate phenotypes that operate within carcinoma cells, and this begins to ask the question of how complicated is the EMT program in actual carcinomas. Can one separate them better? And here, he began to develop a new marker, integrin beta 4, ITGB4 as it's indicated here. Normally, as I showed you before, if one uses the CD44 and CD24 cell surface markers for cracks, one can isolate a population of more mesenchymal cells here, echoing what I showed you in one of the earlier slides. However, if one uses, instead of CD24, now one uses integrin beta 4, then this population of ostensibly homogeneous cells is now resolved into a whole series of intermediate phenotypes that are phenotypically quite heterogeneous. And this begins to suggest that there's great heterogeneity in the activation of the EMT program. Here he developed work which at one point seemed quite counterintuitive, because if he asked for the expression of integrin beta 4, which is an epithelial marker, he found that cells that express integrin beta 4, 1 in 3,000 of these cells could serve as a tumor-initiating cell at limiting dilutions. But if the cells lacked this epithelial marker, now 1 in 750,000 cells could serve as a tumor-initiating cell, a 250-fold difference. This seems exactly opposite of what I'm telling you, what I told you before, because before I argued that the more mesenchymal cells are endowed with tumor-initiating capability. Here I show you exactly the opposite. And this begins to indicate the heterogeneity of the expression of the EMT program. Over here, just to repeat what I said, as cells move from E to M, one has a tumor-initiating frequency of 1 in 3,000. However, as he found, as cells move over here farther to the right, now these more mesenchymal cells have a far lower tumor-initiating capability. And that begins to suggest that there is a program, a succession of changes that happens as cells move from E, fully epithelial, all the way over to M, and that cells moving all the way over to M over here actually lose tumor-initiating capability, causing him to argue, correctly I believe, that in fact the cancer stem cells reside in the middle. That is to say, they're quasi-epithelial, quasi-mesenchymal. And therefore cells that have gone all the way over here actually lose tumor-initiating capability, which explains what I showed you just before. And therefore there have been publications which say proudly that if they, in certain laboratories, they activated the EMT program and they lost tumor-initiating capability, now one can understand that they lost it, and they would conclude that the EMT program has nothing to do with cancer stem cells, but here one can begin to conclude that they pushed cells too far through the EMT program, where they lost tumor-initiating capability, where here one sees the cancer stem cells somewhere in the middle, and we don't know precisely where they are, and indeed we've never isolated pure populations of cancer stem cells, so we don't know all the details of their phenotype. Importantly, spontaneously arising carcinoma cells, to my knowledge, ever complete a full EMT program. The notion that one can move from a fully epithelial state to a fully mesenchymal state is largely an experimental artifact. And when the EMT program is activated spontaneously in tissues, both non-neoplastic and neoplastic, cancer cells advance to various stages along here, but never go all the way. One can push cells all the way to the mesenchymal state experimentally if one overexpresses constitutively high levels of an EMT-inducing transcription factor. But again, to my mind, this is an experimental artifact, and cancer cells that activate the EMT program go over to here. So you can see there's a whole series of changes which occur as cancer cells invade increasingly the mesenchymal direction, but never complete the journey all the way to the fully mesenchymal state. Here, by the way, is integrin beta 4, the marker that I described to you before. Here one can see actually the results of Brian Beery's work, where he found that cells that express high levels of integrin beta 4 and therefore reside over here, such that women who have carcinomas with high levels of integrin beta 4 have lower relapse-free survival in the clinic. Here one sees it into two different subtypes of breast cancer. So women who express high levels of this epithelial marker, we would say paradoxically, have actually have shorter survival because their carcinoma cells seem to reside in the midway between the epithelial and mesenchymal state. Here is the way this has been depicted by Angela Nieto in Spain, where one starts here with the epithelial state. Here's mesenchymal, and she depicted here some years ago a whole series of intermediate states, with the implication being that carcinoma cells can dwell quasi-stably or metastably in intermediate states on the road to becoming fully mesenchymal. And this model is basically correct, we believe, but we don't know, as I'll show you momentarily, how many intermediate states there are, and importantly, how rapidly cells can interchange between residing in one state or another state, that is to say, the phenotype of phenotypic plasticity. Here's yet another manifestation of this, where as one cells move from epithelial to fully mesenchymal, they become increasingly invasive. They acquire increasing drug resistance, I showed you that, and the tumor initiating ability, as I also showed you, resides somewhere in the middle. And so this also has implications, this plasticity, for how one can treat cancers. I won't show you the evidence, but I'll just give you the conclusion for this. Ideally, if one could, one would develop a therapy which does not yet exist to eliminate the more mesenchymal cancer stem cells. And if one were to do that, then ideally, in a theoretical world, one has a situation like this in a normal tumor, where here are the cancer stem cells. If one successfully and selectively eliminated the cancer stem cells, then in principle, the tumor as a whole should eventually disappear because its sources of self-renewal would have been eliminated. But in the real world, that doesn't work because, as we found some years ago, if you eliminate the cancer stem cells, the non-stem cells can actually spontaneously regenerate a new population of cancer stem cells. That is to say, can de-differentiate in a more mesenchymal direction, this representing the phenotype of plasticity that I mentioned to you before. And this complicates the design of future anti-cancer therapies simply because it dictates that one must eliminate both the cancer stem cells and the non-stem cells if one aspires to getting a truly durable clinical response. So here's the work of Yin Zhang, who began to ask the question, what controls the stability of cells residing in one of these states or another? And so he began to resolve these experimentally transformed human mammary epithelial cells into 3 states. Here's CD44, and this is the same as integrin beta 4 here, using the aforementioned integrin beta 4 as a marker on the x-axis. And here he was able to resolve 3 distinct populations of cells. Here again are the fully mesenchymal cells, down here are the more epithelial cells, and here are cells in the middle, suggesting at least 3 distinct phenotypic states, although I will argue there are many more in between that are not resolved by this type of analysis. And the question is, and here you can see the morphology of these cells in monolayer culture. So the question is, could he isolate fully epithelial cell clones from here using FACS? Again, CD104 is the same as integrin beta 4, and observe their behavior. And indeed, he succeeded in doing so. Here is one clone of cells. It's highly epithelial. It's in the lower right. And when he propagated it for several weeks subsequently, these cells remain in the epithelial state. This is what they looked like several weeks later. If you looked at this clone of cells, these cells regenerated both intermediate cells, mixed epithelial mesenchymal cells, and highly mesenchymal cells. And this clone of cells went halfway and created quasi-mesenchymal cells. So the phenotypes of these 3 subclones of cells from this originally mixed population were quite different with respect to their ability to maintain stably their residence in the pre-existing epithelial state, which is, as I say, down here. And then he began to ask the question about what maintains this. And here is the work that he undertook with Matan Hofri and Aviv Beger, the names are familiar to you most likely, in which he began to ask how many distinct transcriptomes are there in these mixed populations of cells. And this is only a first analysis, but you can begin to see here from isolating clusters of cells that have distinct transcriptomes that there begins to seem to be at least 4, 5, 6 distinct transcriptional states intervening between the epithelial and the mesenchymal state, already beginning to suggest that it is not a continuum, but that there are distinct quantal states, almost like the electrons in an atom. But I will not pursue this further. So he began to ask the question, what about the epithelial cells that remain here? What maintains them in the epithelial state fully? Or, to put it another way, can one destabilize their residence in the epithelial state? And to do so, he began to characterize them biologically. The fully epithelial cells, when they were put in three-dimensional culture, and their ability to generate tumor spheres, which are an in vitro surrogate of tumor formation. The fully epithelial cells failed to form any tumor spheres. The cells that were quite plastic and formed mesenchymal derivatives were relatively good. And those that were formed partially mesenchymal derivatives, they were intermediate in their tumor sphere forming potential. In principle, reflecting the ability, and I say in principle, reflecting the ability of these various cell populations to initiate tumors in vivo. And so he began to use CRISPR-Cas9. This is my other attempt at acquiring respectability here on Weizmann, because plus I would come here without talking about CRISPR-Cas9 stimulation. And the question was, what happens if you knocked out specific genes in these epithelial cells using essentially a genome-wide CRISPR-Cas9 library? This is an unbiased screen. What CRISPR knockouts can inactivate a gene, thereby destabilizing the residence of these cells from their previously stable residence in an epithelial state. And there are some experimental challenges here, but Weizmann, so I will continue to proceed, because he began to ask, using various FACS analyses, how one can isolate cells that were previously epithelial and their residence in the pre-existing epithelial state has now been destabilized and they become more mesenchymal because they have lost one or another gene that was required for stable residence in the EMT state. And he used a whole series of small guide RNAs and further bioinformatic filtering. And here, is the list of genes that he came out with, whose expression seems to be critical. Any one of these critical for the stable residence of these cells in the epithelial state. And one can see them here. They're actually quite interesting. Some of them are actually involved in, oval has already been shown to be a guardian against EMT, to protect against EMT. And some of these that he arrived at are parts of this so-called complex transcription factor complex, which is involved in histone modification. These are still early days, but it suggests that he is beginning to resolve the mechanisms by which epithelial cells maintain and stabilize their residence in the epithelial state. Hopefully, if I come back here 1 year, and in Yetze Hashem, I will reach even half the age of Michael Sella and Bedliot, we'll see whether we really understand the molecular mechanisms that are important for maintaining cells in this state. So here's yet another approach at this, because Artie Lambert began to examine what's the nature of the transcription factors that are operating here to maintain stemness. And here he used a whole series of analyses of chromatin, transcriptional output, enhancer landscape, and chromatin accessibility to ask what genes are on in the more stem-like cells versus the non-stem-like cells. And here is an idealized promoter, where here one sees using ChIP-seq that a whole series of transcriptional enhancers are activated in the promoter region of a gene. And this clustering of enhancers is often called a super enhancer, and the transcription factors that localize there appear to drive high-level transcription of a gene downstream of this area here in the promoter. Within this area of super enhancers, where there are multiple transcription factors associated, he began to then look for the open chromatin using the ATAC-seq strategy And here you can see within this super enhancer area, he found, in this case, 4, maybe 5 distinct areas of open chromatin. And he focused on these distinct areas of open chromatin as potentially revealing sites where transcription factors were operating to enable cells to reside in the stem cell state. And what he looked for in these open areas were transcription factor binding sites that are typical of different transcription factors that are known to him. And here what he found was that there certainly were open sites here, but far higher than all the other transcription factors were tumor promoter P63, tumor promoter 73, and TP53. And who am I to talk about P53 here in the presence of Varda Otter and Moshe Owen, or as I often call him, much to its embarrassment, Moshe Rabbeinu, here. Because what one sees here is that these open chromatin regions actually greatly prefer to have these 2 transcription factors associated with them, which are known from earlier work to be involved in the basal stem-like cells in the mammary gland. And in fact, TP63 and 73, they bind directly in the middle of these open chromatin regions. And so, and moreover, as he could show, actually these TP73 and 63, they actually activate their own promoters. Here you can see the manifestation of the super enhancer where there are multiple peaks where one has activated enhancers. And here one can see that 63 and 73 bind in the middle of these open chromosome regions. And in fact, his latest work reveals that 73 is likely to be the master controller of residence in the stem cell state. It turns on 63, and 63 is directly responsible, he believes at this stage, tentatively, for choreographing the stem cell state. This requires us, in the long run, to connect 2 master regulators of stemness, which we still have not done. Because I showed you early on that, in fact, slug is important in normal mammary stemness, and snail is important in the stemness of carcinoma cells. And here I showed you that another set of transcription factors is also important, and I haven't connected the 2 yet, or at least Artie Lambert has not. And we're now examining how these 2 are connected, because both are clearly critical to choreograph, to orchestrate the stem cell state. And here he sees, for example, that if he has cells that are present here in cells that are in the integrin beta high epithelial state, if he knocks out either P63 or 73, then the cells move from a more epithelial to a more mesenchymal state, indicating that 63 and 73 are not only present in these more stem-like cells, but they're also critical to maintaining the residence of these cells in the stem-like state by knocking them out here with small guide RNAs. And so this would be the dream for the future. SE here means super enhancers, and he may be close to being able to draw a wiring diagram of the stem cell state in these cancer stem cells. But that is still, this is still hopeful thinking for the future, because we're not there yet. So אני מודה לכולכם, רב תודות להזמנה, thanks for the invitation. Moshe Rabbeinu, what do I do next?
Okay, we can take a few questions. Anything very burning? Rod?
Oh, so talking about changes, I have to admit the new way of showing science as poetry will introduce the touch.
I'll touch you.
And my question is as follows. You have
I have no aspirations.
Malasot.
We go loud. Okay, anyhow.
So you described a wonderful way of transdifferentiation of epithelial into mesenchymal cells, and this is really sort of a basis for all this transition from being one type to another. And you sort of suggested that middle phases can be associated with reprogramming, hence reprogramming, cell reprogramming.
Well,
Wait, because you have involved
interrupt for a second. Transdifferentiation is usually interpreted as meaning cells move from one differentiation lineage to another one. Here I'm talking about epithelial cells moving in principle reversibly to a less epithelial and then maybe returning to an epithelial state, but not really going into distinct differentiation lineages. I believe there's a different process involved in transdifferentiation as you describe it. You use the word. And the differentiation of embryonic stem cells, in my opinion, has very little to do at all with what I've shown you here today.
So my question is, do you believe that in every epithelial or original tumor, you have already some population which is really of the nature of a cancer stem cell, and all what is happening is really enrichment of this population? Or alternatively, do you believe that there are all kinds of transitions of differentiating cells to stem cells, cancer stem cells, and reverse? So in the middle there is a certain ongoing stemness process.
So I believe, which is different from knowing, that within all tumors there's at least a minor population of stem-like cells. They may be very small in number, less than 1%, but I believe they exist there. Secondly, we've never isolated pure populations of stem cells. And thirdly, I believe all this is different from transdifferentiation going from one lineage to another. How that happens, I wouldn't know. And maybe I didn't even answer your question, but it sounded good, right? We'll have a
Okay, more questions?
The epithelial locked-in state that you showed, do you think that that is also important to have cells that cannot differentiate into leukemous cells?
I'm glad you asked that question. I tell my students, never compliment a questioner by saying it's an excellent question, because somebody else raises their hand and I don't compliment them equally. That other person feels like hurt. It was a very good question. There is evidence that when more mesenchymal carcinoma cells disseminate and seed new tumors, they form tumors better if the more mesenchymal tumor-initiating cell begins in the site of metastasis formation to generate more epithelial derivatives, more differentiated derivatives, so that the hierarchy of phenotypes that operates within a metastatic colony begins to recapitulate the hierarchy or the spectrum of phenotypes that pre-existed in the primary tumor prior to metastatic dissemination. And why one needs a mixture of epithelial cells and mesenchymal cells to form a more robustly growing metastatic colony, we don't understand at the cell biological level. And again, we don't understand what the connection is between the EMT program and stemness. Why is it there? Ainli Shumusag.
So, yeah, you want to ask?
I'm trying to go backwards and see how you trace P73. So where does epigenetics come into it, and what do you think is changed? So I would assume that there would be differential expression of P73 in metastatic and non-metastatic breast cancers, and is that true? And what derives the change in P73 expression that leads back to epigenetics?
Well, first of all, we haven't looked into the representation of P73 in metastatic breast cancer versus non-metastatic breast cancer. It's clearly expressed at elevated levels in stem cells, both normal and neoplastic. And what turns it on, we don't really know. Maybe slug is involved. Maybe slug controls P73 expression. That would be a very nice and simple solution, but so far we have not been able to demonstrate that experimentally. So right now we're working on that upstream circuit, but we still don't really understand how the regulation of that operates.
Any burning questions further? No,
That's
thank you very much, Bob.